Okay, hi guys, welcome to Biology Online with Henny van Vieren. Okay, hello year 12s, welcome to our last um, lesson for this term, believe it or not. So we've made it, 2 weeks of online learning. You have survived. I'm hoping that you actually learned something. I really miss seeing you guys face to face. It's a lot more fun over there. We go on sidetracks and all types of stuff, whereas when I'm here alone at home, it's like, feels like you're talking to yourself a lot. And I know I do that, but that's okay. Anyways, so today's lesson is all about DNA replication, and you will remember that previously we spoke of enzymes and how enzymes work and factors affecting them and so forth. Now, DNA replication is one of those processes. Remember, I said it's like a production line. I showed you some pictures of car, automobile manufacturing facilities and their production lines. So just remember that all the DNA replication that we're going to be talking about now is a enzyme driven thing. It doesn't happen spontaneously. The enzymes are basically produced by the nucleus to then start running this process when it is needed. Okay, so over to the lesson. Alrighty, so we've got replication of DNA, and this is quite a nice little picture of the actual um, double helix as we know it today, all because of two gentlemen called Watson and Crick. Now, look, I made you a little animation. I was obviously bored and was playing around with Photoshop. Um, so when we look at the cell cycle, this is the cell cycle over here, and most of the cell's life is spent in G1. Now, some cells grow slower than others, and consequently the G1 may be a lot more of the cell cycle. It's not like it shows here. Some cells need replacement very quickly, so consequently they will also then have a shorter version of the G1 cycle. Okay, so just keep that in mind, but your major portion of the cell's life is usually spent in G1, and that is growth. So just after mitosis, now the first thing we need to say is mitosis is for growth and repair. Okay, so growth and repair means we need the same cell right there again. We don't suddenly need a cell that codes for cartilage that comes up where our fingernails are supposed to be. Okay, it needs to be, it needs to be the right cell for the right place. But how that goes with growth and repair is that our cells simply duplicate themselves. Okay, now we start off with a little baby cell. It's not really a baby cell because they just duplicated, so it's a new cell, but we call them daughter cells. And this is just after mitosis, right there, okay, at the beginning. So we're just tiny, 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 and look, I made you, it's now turning into a bigger one. Okay, so as it grows, it realizes it basically is in a growth phase. So it grows slowly, it makes more, mitochondria that it needs, it makes more organelles, um, and so forth. And then it gets to a point where it gets really big and old. Look, I even put in a beard for you guys. Haha. Anyways, and now the actual surface area to volume ratio becomes a problem because the cell is now physically so large that to continue growing will mean that we have to expend a lot of energy just moving stuff around um, within our cell itself. But if we divide this thing in two, it's going to take up the same amount of space, but we will have a much better surface area to volume ratio. Does that make sense? So that is why during growth our cells will basically divide, and then as they grow again they will divide again, and so on and so on, and that is how we grow. So when we get to this point, we're at the end of G1. Now the cell goes into S phase. Okay, and the S phase is where we have DNA replication. That is really important that you understand this. The DNA is replicated is duplicated long before the cell actually splits. The cell prepares in advance and basically say, you know what, this is all my data, all my amazing textbooks, it is my reference library. I need to duplicate this before I can split myself in two. Otherwise, I will have a cell that doesn't have the DNA, and consequently that cell will be useless. So it starts duplicating its DNA. And that's what we're going to be looking at. But then during G2, and this is after the DNA has already been duplicated, now it duplicates and still grows things like extra mitochondria. Because as you replicate, and as you then, not replicate, as you divide, then you need enough like mitochondria, ribosomes. If you're a plant, chloroplasts, you need 2 Golgi bodies at least. You need enough endoplasmic reticulum, and so forth for every one of those cells to be a functional cell. I hope that makes sense. So that's G2, and then when it's ready, it goes into mitosis. Okay, and mitosis is for growth and repair. So that is the cell cycle. Now, this is a video regarding mitosis from the amoeba twins. The amoeba sisters. They are amazing. These guys, I cannot sing their praises high enough. It is their video. I'm not taking any ownership of their, their intellectual rights or any of that. But yeah, they are absolutely
Have you ever
brilliant.
looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it and a few days later it's gone. It's all healed and you don't even think anything of this amazing process that causes this to happen.
Band-Aids.
Or let's say you're looking at your nails and you notice they're a lot longer since the last time you cut them. Or let's say you're looking in the mirror and you notice that you are a lot bigger than you were when you were 5 years old.
I'm already a lot bigger than what I was before I started this lockdown.
One major thing they have in common is mitosis. Mitosis is a type of cell division done by most of your body cells, and it's really important for cells to divide. If they didn't divide, you wouldn't grow. I mean, how do you grow if you can't make more cells, right? So one reason why you're bigger than you were when you were 5 is mitosis. Mitosis also is great for repair of damage. If you have some kind of accident, like when we were talking about that cut on your arm or leg, well, you want to make sure it gets repaired well. So you have to make more cells to do that. Mitosis is great for that. Now, it's really important to understand what it is not. Mitosis is not a process that makes sperm or egg cells, because that's something different called meiosis, which sounds like mitosis, unfortunately, but it's a different process.
This is very important.
Mitosis is done to produce body cells. Mitosis makes identical cells. That is the goal, identical cells. So if you're trying to make more skin cells to replace worn out or damaged skin cells, you don't want to start suddenly making stomach cells there. That would be ridiculous. You want to make sure you have identical cells replacing what was lost. So mitosis makes identical cells. It's a really important thing. Now, it's also important to understand that your cells are not dividing all the time. If all they did was divide, it would just be rapid, crazy growth. In fact, this is kind of what cancer is. Cancer is uncontrolled cell growth. We have a clip on the cell cycle and what the cell is usually doing most of its daily life, which is actually a phase called interphase, where it's growing and replicating its DNA and carrying out its daily cell functions. That's where cells spend most of their time in respect to the whole cell cycle. Mitosis is a very short amount of time in respect to the whole cell cycle. But mitosis is a critical process because this is where it is going to divide and make more cells. Before we get into the steps of division, it's really important to understand that your cells have something inside them, an organelle called the nucleus. And the nucleus holds your DNA. DNA is really important because it's your genetic information. And if you're going to make more cells, you need to have the same DNA in those new cells as you did in your original cells. You want it to be identical, no mistakes. Very important. The problem is you've got a lot of DNA, and we've got to get that DNA into the new cells using mitosis. So there has to be a better way to organize that DNA. Well, what actually happens is that DNA can be organized into these condensed units called chromosomes. Chromosomes are made of DNA and protein. You've probably heard before that humans have 46 chromosomes. That means 46 chromosomes are found in most human body cell nuclei. What are nuclei? Well, it's the plural of nucleus. You don't say nucleuses, you say nuclei. Well, in the nuclei, there are 46 chromosomes. Organizing DNA into condensed chromosomes makes it a lot easier to move over when you're making new cells. So if you have 46 chromosomes in a human body cell, you have to duplicate those chromosomes in interphase before mitosis starts. That basically means you're duplicating your DNA, since chromosomes are made of DNA and protein. You have to do this before mitosis starts because if you're going to make an identical cell that has 46 chromosomes just like the original, well, it makes sense you have to duplicate the genetic material before splitting. So if you look at our cell cycle video clip, we talk about interphase. That's a stage where most of the time cells are spending their time. They're actually duplicating their DNA during that time. So ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes.
This is really important.
As the sister chromatids are...
So normally when you're in G1 phase and the cell is just growing, your chromosomes are only in this single shape, okay? This is called a chromatid, and with the centromere, it is called a chromosome. Now, when it then goes into the S phase, when it duplicates itself, it's still connected. So it's still the same chromosome, but they are direct copies of each other. So you will have the gene for blue eye color there and the gene for blue eye color there. It's exactly the same. Okay, so all they did is they duplicated the DNA in preparation for mitosis. They are not going to separate before they go into mitosis. So from S phase, when they are finished duplicating, until they get to the end of G2 and start mitosis, this is how they look. But for the longest time, they are a single chromosome, a single chromatid.
Still attached, and we're counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture, but you went from 46 to 92 chromatids. We have a video explaining that in more depth and how that factors in for mitosis. Okay, so now we can get right into mitosis. I like to tell students to remember
You don't need to know these
PMAT.
names, okay?
It's a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and the T is for telophase. So remember PMAT, the stages in order. The very first step is prophase. Prophase because it's the beginning step, the nucleus is still there.
Yeah, just the word PMAT reminds me of the mat that's in front of a toilet, especially when you go and visit some old people. That's weird. Anyways.
And it's going to go away later on, but this is a stage where it's actually still there. The chromosomes are visible. In fact, we say they're condensing, which means they are thickening and visible. The next stage is metaphase. M for metaphase, but I also like to remember M for middle because in this stage, the chromosomes line up in the middle of the cell. The nucleus has been disassembled, it's no longer there, so we've got the chromosomes in the middle waiting there.
So the membrane is gone.
Next, the A is for anaphase. In anaphase, I like to think as the A for away. The chromosomes move away. They are moving to opposite sides of the cell, so they are moving towards the poles of the cell. Now, one thing to point out, these chromosomes, they're not moving by themselves. They actually have something called spindles. These spindles are fibers that help move the chromosomes to the ends. Kind of helps them move along. The last stage of mitosis, think T for telophase, In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. I like to think of the T for 2 because you can really see in this step that the end goal is going to be 2 cells, and in the human body they're each going to have 46 chromosomes. And again, remember, they are identical. Now, cytokinesis is responsible for the final separation into 2 cells by splitting the cytoplasm, which completes after the PMAT mitosis stages. So why did all this matter? Without understanding cell division, we wouldn't understand how growth and repair happens, because they both require more cells to be made. Understanding mitosis is also very important for cancer research. Cancer itself is uncontrolled cell growth. So, in other words, uncontrolled mitosis. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So seriously, guys, I can't sing their praises high enough. They are amazing. So just going on, we need to look at the details of DNA. And DNA, as you know from year 10 and year 11, DNA is the abbreviation for deoxyribonucleic acid. Deoxy means it's lost in oxygen. Ribo refers to ribose sugar, and then the nucleic acid. Now this is the information carrier. We've talked about this many times. I mentioned to you that in every single one of your trillion cells, you've got about 700 megabytes of data on that molecule, and that molecule is between 5 and 7 feet long, depending on who you ask. Well, if you put all the molecules together, basically. And in that is the instruction for you and everything that makes you. And it is completely unique. So it's DNA, the way that they package it. If you think about a ribbon, they take the ribbon and we start twisting it and twisting it and twisting it and twisting it and twisting it, and eventually your ribbon gets shorter and shorter and shorter and shorter. And then you take that short version and you put it around a yo-yo, and this yo-yo is called a histone. And then we take those histones and we put them into a chain, and eventually, I'll show you another picture a little bit later, they become the chromosomes. So chromosomes are twisted and twisted and twisted DNA. Okay, so again, I mentioned earlier during the video that when we look at this is the human karyotype. Okay, now for those of you who've looked at this before, is this a boy or a girl? It is a boy. The reason why it's a boy, it's got the Y chromosome. Now, we have 46 chromosomes, or 23 pairs of chromosomes. These are not the same as the X-shape double chromatids. So just before it goes through mitosis, all of these, irrespective of which pair they are, will form that X shape with itself to duplicate. So this one, that's not good. Give me a highlighter, please. And I want blue. So this one over here is gonna create another blue one, and the reason why I'm drawing it blue is because this one was maybe from the dad, okay, and there's the centrum here, also in blue. And then when you go to the other one, and this one would have been from maternal origin, so there's not really a pink here, but let's use, oh wait, there was. Oh no, there's not. See, I am colorblind. Anyways, but this one over here will look like that. So every single one of these will duplicate themselves before, I wish they wouldn't do that, but before they go through mitosis. Okay, so in most of G phase, the DNA look like this. They are singles, and then single chromatids is a single chromosome, but they are in the homologous pair. So homologous means the same. Okay, so this one was from your mom, and this one was from your dad. This one contains the gene for hair color, and this one contains the gene for hair color. But this gene for hair color is the recipe from your mom's family side, and this gene for hair color is the recipe from your dad's side. Just like this might be the gene, the recipe for your dad's lasagna, and this might be the recipe for your mom's lasagna. Your mom puts spinach in hers and so forth, and your dad puts eggplant in his, but they still make lasagna. So, and we'll talk about that later, which one is expressed and which one is not. But they are called homologous because they contain the same genes, but different versions of that same gene. Okay, alrighty, so homologous pairs 23. D for diploid, okay? When you are diploid, you have 23 pairs or 46 chromosomes. When you are haploid, you only have 23. You don't have 23 pairs anymore. But that is important when we get to meiosis, not during mitosis. You can see there, this is prior to mitosis. And as you can see, they all duplicated themselves. So we have replicated DNA here, and they are ready to enter mitosis. Okay, so here we are. You start off with the ribbon. The backbone on the edge of this ribbon is formed by ribose, sugar, and phosphate. So we call it the sugar phosphate backbone. And this is on both sides. And then you have the nucleotides. Adenine, remember we talked about ATP, adenosine triphosphate. It's the same thing, it only has 3 phosphates here. But adenine binds to thymine. Now, I want you to please always remember that adenine and thymine, and they have 2. Guanine and cytosine, they will always be complementary to each other, and they go to there. So the easy way to remember it is, have you ever been to the Gold Coast in Australia? You say at the Gold Coast. And if you remember at the Gold Coast, then you will remember this. The other way that I remember it is, if you look at an A and you look at a T, both of them contain only straight lines. And in fact, if I go and take my A and my T, I can kind of put them together. Okay? But then I go and I say guanine and cytosine. Both of them start off with a C shape. But then to turn it into the, so very curvy, okay? But to turn it into the G for guanine, all I do is I do that. So guanine and cytosine have the curves, and they have 3 bindings, okay? Adenine and thymine, AT, they have the straight lines, okay? At the Gold Coast. Please remember that, very important. Okay, so now we wind up this ribbon. And as it is wound up quite tightly, we then wind it around this histone, almost like you're winding up a yo-yo. And then the histones are wound up. This chain of histones are wound up, and eventually everything gets wound up again until you form a chromosome. Alright, and the chromosomes are stored in the nucleus. So I hope that makes sense. Now I think this lesson has been long enough. I'm going to park it here. When we come back next term, we will have at least hopefully 3 weeks to complete this. Yeah, so before we go into DNA replication, if you do want to go on yourself, this video explains it really nicely. Here's another one that explains it nicely. Yeah, but I will post the video. I don't expect you guys to work during the holidays. If you do want to, you're obviously more than welcome to. Okay, So for the moment, that is the basic structure. Thank you guys. Please look after yourselves during the holiday. Stay safe and enjoy. I'll see you soon. Bye.